and you all will have 6 minutes to check out the warm up per usual. We started a new module. That link should be active. So if you click on the modules button on your home screen, it should take you directly to our Unit 3 module. Of course, today is the first day of Unit 3, so you'll have 6 minutes to knock out the Unit 3 Day 1 warm up. All right, so hopefully that was enough time to complete the warm-up. 6 minutes, we've got a lot to cover today, so I don't really want to waste any time waiting for people. Thank you all for being here and being here on time. Keep in mind that all of our assignments for this unit will be due next Wednesday. Next Wednesday is going to be the last day of Unit 3, so you've got until February 10th to knock out these assignments. What you'll notice is that the warm-ups and exit tickets this unit are going to perfectly mirror the types of questions that you'll see on the exam, which will be next Wednesday. So I really encourage you all to do them. You have unlimited attempts to do them. So if you're studying for the exam next week or over the weekend, you can go back and do your warm-ups and exit tickets. That's a great way of practicing the types of questions you'll see on the exam, because I literally took them from the same source where I get the exam questions. So please be using those strategically. Please make sure that you're doing them as you go. So the objective today is to analyze how cells grow and reproduce in terms of the cell cycle. We'll learn exactly what that is, but this cell cycle has 3 parts: interphase, mitosis, and cytokinesis. The essential question today is why and how do cells reproduce? So we seek to answer that essential question by the end of the lesson. All of the notes that you're taking, and yes, you should be taking notes, should be in support of that essential question. So this is just a reminder, have your notebook out, have your pen or pencil ready, be ready to take some strong notes. Today's lesson is titled The Cell Cycle Overview. Today is February 2nd, 2021, and it is Unit 3, Day 1. All right. So before we jump into the content, we've got to celebrate our 4 classmates who mastered the exam yesterday. I want to give them a round of applause. You guys can feel free to send them some nice congratulatory messages in the chat. I think that would make us all feel good. But Fraylin, Rudy, Cindy, and Kathy, you all performed amazingly yesterday. You're off to a great start as we progress through this biology class. Unit 2 is typically a unit where students do perform better, and we're going to look at some of the data in a couple of slides. But I am still extremely proud of these 4 students who earned greater than an 80% before the curve. Okay, so these students did an excellent job. We want to congratulate them. For those of you who weren't there this time and may have been close, keep pushing. I expect that everybody will be at mastery by the end of the class. Remember, our ultimate goal is to master the EOC exam in May. So this is by no means an opportunity to feel discouraged. If you weren't there, you will get there. Keep working hard. Keep scheduling appointments with me. That being said, I'm going to send a link to schedule appointments with me for one-on-one tutoring to the chat right now. And I just wanted to show these students some praise. So I appreciate Brianna for sending that nice message in the chat. Additionally, we had 8 students, unfortunately, who did not submit the exam yesterday. So if you see your name on the screen right now, we need to get that exam submitted today. We can do that after school. I'm happy to sit with you, but of course I do need to proctor the exam, via Google Meets. So we need to find a time to do that. I can't just give you the code to take it on your own time. So Brian, Pedro, Chandler, Ayana, Tatiana, Christian, Michaela, Kadaria, you guys all need to meet with me so that we can make sure we get this exam into the grade book ASAP. I'll leave this up so you can take note of your name if you see it. Thanks for joining us, Sade. I see you've just joined. And I'll send that link one more time. So you can just go to this Calendly link and it will allow you to schedule a time with me. It will force you to do it in only 15-minute blocks, but of course it will take you longer than that to do the test. We can be longer than that, so no big deal. All right, and then we just want to take a look at some of the data from not only this exam, but also from our previous exam as well, and do some comparisons. So we can see that we had fewer test takers. We still got some folks, as we just saw on the last slide, who do need to take this exam. Keep in mind, this is for all of my classes. So there's another 3rd block class that is an honors class. We can see that our average improved. So that's something that we can be happy about. In our first unit test, we had an average of 53.2%. This unit test, we're up to a 56.78%. That's not where we want to be. Obviously, we want to be at a mastery level, which is an 80% or above, but we're seeing improvement, and we will continue to see that improvement as we get more exposure to these standards and do some more practice. We can also see that in this unit, we had a big increase in the number of students who earned A's and B's on the exam. We went from 9 to 13. So 40% of students who have taken the test so far earned an A or a B. That's excellent. That's something that I would love to continue to see growing. I think we will continue to see that growing. And I want to acknowledge that, highlight that. So there's reason for us to be encouraged here. We're seeing growth. We're seeing people improve, and it will only get better. We've just got to continue to work hard. We've got to get some of these assignments submitted. We have a lot of missing assignments in this class. We need to get those assignments submitted, and that will only boost your performance further. All right, now we're going to jump into the content. We know that Sternger is a concept that we've introduced since Unit 0. It's a concept that we have to keep coming back to, and we will weave it throughout our course. Can somebody recall for me what the R's in Sternger stand for? We've got 3 R's. Can anybody remember one of them? Reproduction. Wonderful. And that's the exact one I'm looking for, Jason. So you hit the nail on the head. This unit, we're going to focus specifically on that second to last R, which stands for reproduction. The reproduction of offspring by a sexual or asexual process. Organisms do this in order to make new cells or in order to copy/preserve their DNA. Almost all living cells will reproduce in their lifetime. Okay. Abraham, I see you threw in another R, regulation. That's excellent. We will talk about regulation more so in the next unit. So in this unit, we want to focus on reproduction. All right, now we're jumping into it. For my active note takers, which should be everybody, you're seeing that we have words that are highlighted. These words that are highlighted in yellow, or if your preferred language is Spanish, in green, should be written down in your notebook. All right, so let's focus on these highlighted words. Let's get them written down. Would anybody like to read the definition of cell division?
I can read it.
Thank you so much. Go ahead, Brianna.
Cell division is a process by which individual cells and unicellular organisms reproduce.
Wonderful. Thank you so much. So we're going to talk about this process of cell division. That phrase, cell division, is basically what's the word I'm looking for? It's a synonym of the word reproduction. All right, so we use those two phrases interchangeably. In this sense, when we're talking about reproduction, what we're talking about is cell division.
Oh, can you go back?
Yes, I'm sorry, I didn't move. I didn't mean to do that. Okay. So here's an image of cell division actually taking place and through a microscopic image. It's a plant cell on the left. We're going to talk about what exactly we're seeing there because we can see some neon orange substances being pulled apart. We're going to talk about exactly what's happening in that image. And then on the right, we can see bacterial cells reproducing. And so a colony of bacteria can start off as 1 or 2 cells and very quickly become thousands of bacterial cells. And that's what you're seeing in the image on the right. 1 cell divides and becomes 2 cells. 2 cells divide and become 4 cells. 4 cells divide and become 8 cells. So we're seeing exponential growth, and it happens rapidly. So now I have a question for you all. I want you all to think about it. Share it out into the chat, or you can unmute yourself and share it that way. Why are new cells created? What's the whole point? Why do organisms need to create new cells? I've got an answer from Abraham in the chat. He says to replace the old ones. That's definitely one of the primary reasons. Can we think of any others? Thank you, Abraham, for that. Why else might organisms need to create new cells? It's always good to come back to a human context. So think about how humans are born versus how humans end up around the age of 15, 16. What's happening in that timeline? Thank you, Abraham. Yeah, we're growing. That's the other primary reason. Growth needs to take place. In most species of animals, plants, fungi, growth is always happening. So creating new cells provides for an opportunity of growth. And there's a third reason that we'll talk about in another slide, but Abraham got 2 of the 3 there, so that's excellent. And then the second question asks, how are new cells created? That's going to be what we talk about throughout this entire unit. So for the next 8 days or so, that's going to be the focus of this unit. How are new cells created? So we've got some good thoughts. Thank you, Abraham. We're going to build off of those thoughts. This is a cool image that shows that your body is constantly replacing and rebuilding itself. It's creating new cells. So for those of us who are interested potentially in a career in medicine, nursing, pathology, we need to be aware of these different timelines. Skin cells replace themselves very quickly within a month. And that's why you have, if you end up getting a scratch or a cut, it doesn't remain there forever. New cells are going to come replace those damaged cells. Blood is replacing itself every 4 months or so. So that's why it's important that you stay hydrated. It's important that you're getting fresh air. Your blood needs these things to healthily replace itself. And then the liver as well is one of the most prominent organs that can regenerate. So a damaged liver can actually be replaced over a period of time. All right, so let's flash back to Unit 1. This wasn't long ago. It was only 2 weeks ago. So we should remember. Which of our 4 macromolecules is DNA? Which of the 4 macromolecules is DNA?
Want to be the nucleic acid?
Wonderful. Thank you, Abraham. Yeah. So DNA is a nucleic acid. Now, let's hear from someone else. Abraham, you're doing a great job. Thank you for jumping in and contributing to class today. But let's hear from another voice. What is the monomer of a nucleic acid? What's the building block of a nucleic acid? So, Jason, I see your answer in the chat. We've got 4 macromolecules. And Breon, I see your answer. You've got the correct answer, nucleotides. But let me address Jason's answer first. So we've got 4 macromolecules. Those are the polymers. Those are the bigger structures. Nucleic acids are one. Proteins are another one of those polymers. They're another macromolecule. So each of those polymers is made up of smaller building blocks. In the case of nucleic acids, Breon has the correct answer. Nucleotides are the smaller building blocks. And you might hear us refer to them as monomers. You might hear us refer to them as building blocks and maybe even subunits as well. But these nucleotides, when they get chained together, they are actually what is creating a nucleic acid. So we want to come back to that. Of course, this is a cumulative course. We're not going to ever get away from what we learned in Unit 1 and Unit 2. So we'll keep coming back to these concepts. But thank you for that, Abraham, Jason, and Breon. I appreciate your contributions. Good teamwork. All right. So just as another throwback, we did talk about DNA in our first unit, but we're introducing some new terms in this unit. The following terms are synonymous with DNA and genetic information. but they have important differences. And we're going to go into some of these differences over the next few slides. We've got some words here. I want you all to notice the similarities between these terms. They all begin with CHROM, chrome, chromatin, sister chromatids, chromosomes, chromosomes. So these are words that are obviously referring to the same thing. They're referring to DNA. So I want us to have these mental cues. When we see similar words that sound alike, we should be thinking about their similar properties. They're all referring to genetic information. They're referring to DNA. Keep in mind that what you're seeing highlighted in yellow should be being written down in your notebooks. So here, sorry, it's kind of at the bottom being cut off a little bit, but it'll go away eventually. Chromatin is random and disorganized DNA. It looks like a bowl of spaghetti on the left side. So the DNA that exists in a nucleus typically exists as chromatin. It's random, disorganized DNA. To me, it looks like a bowl of spaghetti. Chromosomes are condensed and organized DNA. We can see them on the right side. They are a DNA that is tightly wound up. All right, so again, we want to make sure that we're getting these highlighted words and phrases and sentences written down somewhere in our notebooks. So there's a really important difference between chromatin and chromosomes. Chromatin is the relaxed, random, disorganized DNA. Chromosomes, tightly wound, condensed, organized DNA. This difference will become more important as we talk about some of these processes of cell division. Okay. So these phrases are not highlighted, but they are inside of a yellow box. So I would ask you all to write them down. So I'm going to leave this on the screen for probably 2 minutes or so to give you plenty of time to copy down these definitions. Telomeres. Telomeres. Telomeres are the ends of a chromosome that protect the chromosome from deterioration and fusion. So we can see telomeres at the end. They're represented in the red region here. They're important for protection. Sister chromatids, so there's that chrome again, C-H-R-O-M, sister chromatids are the identical copies of DNA. So each chromosome is made of what looks like an X. Each half of that X is a sister chromatid. And those sister chromatids are connected in the middle in a region that's called the centromere. So get these phrases written down and defined in your notebooks. I'll leave this on the screen for about another 60 seconds to give you all plenty of time. Okay. So we're going to zoom in. We've talked about the cell in our previous unit. We talked about different types of cells. We know that eukaryotic cells have nuclei. They have a nucleus, and that nucleus contains the genetic information, and it controls all of the cell's activities. So the cell, if we zoom in and we look inside of the cell's nucleus, that's where we will find the genetic material. The genetic material is stored in a nucleus. Okay. The genetic material inside of the nucleus is organized in chromosomes. Remember, chromosomes are more organized DNA, and chromatin is disorganized DNA. So inside of the nucleus, the chromosomes are more organized versions of DNA. So we've zoomed in further. We looked inside of the nucleus. All right. And then chromosomes themselves are just tightly wound DNA. Like imagine we had a shoelace and I just wrapped it really, really tightly, hundreds and thousands of times around my finger. I could still unwound it to end up with the long strand, the shoelace itself. I can unwind it to end up with the shoelace, and that shoelace would be the DNA. So the DNA gets really, really tightly wound around itself and around some proteins in the form of DNA to create a chromosome. We'll talk more about this a little bit later in the course, tomorrow actually, but humans have 46 chromosomes. They exist in 23 pairs. You get 23 from your mother and 23 from your father. We'll talk more about that tomorrow. So we've zoomed in. A eukaryotic cell has a nucleus. Inside of the nucleus, we find chromosomes, and chromosomes are made up of tightly wound DNA. So nothing to write on this slide. I just want to make you aware of the fact that all of your body's cells are constantly dividing. Not all, most of your body's cells are constantly dividing. And again, Abraham gave us two solid reasons as to why this is happening: to replace the old cells and to grow. As we'll see in the next few slides, there is another reason that's quite important as well. In general, there are two cell types. There are gametes, which are sex cells, and there are somatic cells. So your gametes are your sperm and your egg cells. They get made by a special process that we'll talk more about on Thursday. All the other cells in your body are called somatic cells. In fact, soma, S-O-M-A, is a root word that means body. So your somatic cells are all the other cells that make you up. Your nerve cells, your muscle cells, your digestive cells, your skeletal cells, your skin cells. All of the cells that are not sperm or egg cells are somatic cells. Gametes get made by a special process. We'll talk about this process on Thursday. Your other somatic cells get made by a process called mitosis, which we'll talk about tomorrow. So going back to that question, one of the essential questions for today's lesson, why are cells made? Abraham gave us two of the three reasons. He said, replace the old one. Yeah, that's one of the primary reasons. Old cells die. This is a natural process. It's a good thing. Cells need to die off at some point because they start to become less able to do their jobs. And once those cells die, they must be replaced. So replacement is one of the reasons why cells are made. Another, growth. This was the other reason that Abraham gave us. Most organisms grow in size over time. Doesn't matter if it's a fungus, if it's a plant, if it's a jellyfish, if it's a human being. Most organisms grow in size from birth until they reach a stage of adulthood. Typically, they grow until they can reproduce themselves. So growth is the second reason why cells are made. And then the third reason is reproduction, to actually have the ability to mate, to create offspring. Of course, there are two types of reproduction. There is sexual reproduction and there is asexual reproduction. Does anybody know the difference between the two? What's the difference between sexual and asexual reproduction? Or one of the differences? Again, the question is, what is a difference between sexual and asexual reproduction? Okay, thank you, Jason, for acknowledging that I asked a question. Jason says he doesn't have a clue. Anybody else have any thoughts? Okay, so this is something that we haven't learned before. No big deal. We'll talk more about it later on in the course. Sexual reproduction, and I'm typing a message out to the chat. Oh, look, Brianna. Wonderful. Brianna says that asexual reproduction produces offspring that are genetically identical to the one parent. A lot packed into that phrase. I'm going to break it down. So asexual reproduction involves only one parent. This is why it's called asexual. It's only one individual producing the offspring, and that one individual is making an exact copy of itself. Which means that its offspring is going to be genetically identical to the parent. Essentially, it's creating its own identical twin, but not even, it's even more genetically identical than a twin. It is exactly a copy of itself. Typically, go ahead, Brianna.
Okay, so you know how you said there's 23 pairs? Like, what if there is more chromosome from one parent than the other?
Okay, great question. So all human beings, so any organism from a particular species should have the same number of chromosomes as any other organism from that same species. So all human beings, in theory, should have 46 chromosomes. There are some human beings who might have an additional one or two, or they might be missing one or two. Typically, those individuals are not able to reproduce for that very reason that you just brought up. So that was a really insightful and smart question. When two organisms do not have the same number of chromosomes, it becomes far less likely that they can reproduce. And even if they do reproduce, typically their offspring cannot reproduce. So think about this. And this will actually be one of the questions that you all will see in this unit. A wolf is very similar to your dog, right? So if you've got a German Shepherd at home, that German Shepherd is genetically similar to a gray wolf. However, a gray wolf has, let's call it 78 chromosomes, and your German Shepherd has 76. So they're close in number. They can actually reproduce. They can create an offspring. But that offspring is not going to be able to reproduce. It will be sterile because its number of chromosomes has been thrown off. All right, so that was a really, really smart question, Brianna. And this is why organisms like human beings well, let me not use human beings. This is why organisms like giraffes and hippos cannot reproduce. They have different numbers of chromosomes on top of there being other, on top of there being other barriers to reproducing. But genetically, it just is not going to fit. So that was a good question. Jason, to answer your question, is asexual male or female? Typically, organisms that can reproduce asexually, we wouldn't assign them a specific sex or gender. We wouldn't consider them to be male or female. Most organisms that reproduce asexually are unicellular organisms. They're bacteria or they're protists. So they're just making copies of themselves. We don't really consider them having sexes. There are some examples in nature of animals reproducing asexually. For example, some jellyfish can reproduce using their own gametes. And remember, we just talked about a gamete a few slides ago. Let me go back. So some species of jellyfish have their eggs and their own sperm, and they can use both their own sperm and their own eggs to reproduce by themselves. So that's a very unique format or a very unique circumstance that we can sometimes see in nature, but even that is not really asexual reproduction. That would be still sexual reproduction. It's just happening with one individual. It's still sexual because you're still using two different gametes. You're using a sperm and an egg. That is pretty cool. Yeah, jellyfish are really, really, they're animals, but they're not in the animal kingdom. They have their own kingdom. So they have a lot of they do things very differently than most animals do. All right, but good talks, good questions. And Brianna, I'm really happy that you asked that question. That was really, really thoughtful. And Monet, let me address what you said too. She says sexual reproduction just means combining genetic material from two parents. Perfect, yeah. Sexual reproduction typically requires two parents. Like I just said, jellyfish are weird. They do things differently. But in the animal kingdom, it requires two parents. One parent who provides the sperm, one parent who provides the egg. All right, let's see. So this is a video showing dividing cells under a microscope. And you're going to continue to see the cells divide more and more. So we just split into two new cells. There it is again. Tomorrow we'll talk about the actual phases that these cells are going through as they divide. But let's move on. So the actual purpose of this lesson is an overview of the cell cycle. So we've gotten the basic information, the background information that we need in order to now start talking about the cell cycle. Would anybody like to read the definition of the cell cycle? It's written at the top of the page, highlighted in yellow.
I can read it.
Thank you, Brianna.
Cell cycle, the process cells go through to make new cells.
Wonderful. So this process has three stages. The first stage we classify as interphase. The second is called mitosis, and the last is called cytokinesis. Now, interphase can further be broken down into its own 3 stages. There's G1, which stands for growth, S, which stands for synthesis, DNA synthesis. We're making new DNA. And then G2, which also stands for growth. We can see that interphase is clearly the longest part of the cell cycle. G1, S, and G2 make up the majority of the cell cycle. So from this slide, we really just need to have the definition of the cell cycle written. The process, as Brianna said, that cells go through to make new cells. And here are those 3 major parts of the cell cycle. You do need to have these written down. We will talk about them in depth over the next few slides, but please make sure you have them written down in order. First, the cell goes through interphase. Interphase is extremely important because the cell has to copy its DNA before it can divide. I'll say that one more time. The cell has to copy its DNA before it can divide. So let's say that we've got a bacterial cell that normally has 11 chromosomes. Before it can divide, it has to copy itself, copy its DNA to give it 22 chromosomes. That way, when it divides, each of the resulting cells will have the correct number. Each of the resulting cells will have 11. If the cell were to divide before it copied its DNA, we'd end up with 5 chromosomes in one cell and 6 in the other. Neither one of those would have the correct number. Neither one of them has 11. So we've got to copy the DNA first, then the cell can divide. And the copy of the DNA happens in interphase. Cell division happens in M phase, and this could be mitosis or meiosis, 2 different processes that we'll talk about over the next couple of days. And then the third step is cytokinesis. We'll talk about what that means. Cytokinesis. So that first phase of the cell cycle is called interphase. Interphase is the longest phase of the cell cycle. And this is when the cell is preparing itself to divide. There are 3 steps of interphase. There's G1, which is when the cell is growing. That's what the G stands for. The cell is growing. It's also making copies of its vacuoles and its mitochondria and its chloroplasts. It's growing, it's preparing itself to divide. The second part of interphase is called synthesis, and this is when you actually replicate the DNA. You make an exact copy of the DNA. Super important. And then again, it goes through another period of growth in G2 as it continues to prepare itself for division. Now, this is happening rapidly. This isn't happening over the course of days. This is happening over the course of, seconds, as we just saw in that previous video. I can't emphasize enough how important it is that we're taking notes, guys. We're going over a lot of really important stuff. We need to make sure that we've gotten things written down. Okay. We'll talk more about this process of DNA replication, and if this is something that really interests you, I would encourage you to take the AP biology class, challenge yourself next year. We talk a lot about it in depth in AP biology, but during replication, the DNA is unwound. It's kind of like a zipper. So when the DNA is totally packed up, it's totally zipped up, and then it gets unwound so that we can make copies of both sides of the jacket, both sides of the DNA. I need to copy this side and I need to copy this side. And then eventually I end up creating two totally new strands of DNA, but they are copies of one another. They're complementary to one another. So during DNA replication, the DNA is unwound and copied, making two strands from one strand. Okay, seems like we've got a good amount to write on this slide. M phase comes after interphase. M phase is when the chromosomes in the nucleus divide. This is the process of cell division. And there are two types of cell division. One of them is called mitosis. Mitosis is used for asexual reproduction in your somatic cells. Remember, your somatic cells are all the cells in your body except for sperm and egg cells. So all of the cells in your body except for sperm and egg cells use mitosis to divide, as well as unicellular organisms. Meiosis is used for sexual reproduction to create gametes. Meiosis is the process that creates sperm cells and that creates egg cells. We're almost done, folks. I know it's been a long lesson. I told you we had a lot to cover, but it's 11:11, so you can make a wish and wish that this lesson will finish soon. All right, so this is what mitosis essentially looks like. And again, we're going to dedicate tomorrow's lesson to talking about mitosis, but it's kind of like the development or the evolution of the iPhone. Started off with one simple camera. That simple camera ended up having now two lenses, but they were merged together. And now with the most recent iPhones, they're totally separated. And some of them actually have three. But we can see how mitosis takes place. Start off as one, they start to split apart, and eventually you end up with two distinct cameras. That's what mitosis basically looks like. We'll see that tomorrow. And the last phase of the cell cycle is called cytokinesis. Cytokinesis literally means cell movement. Cyto, C-Y-T-O, that means cell. Kinesis means movement. So cytokinesis means cell movement. Some of you might go on to maybe try to study kinesiology if you're interested in being a personal trainer or an athletic trainer. You'll likely have classes in kinesiology, which is the study of how the body moves, how different joints and tendons and ligaments allow the body to move in specific ways. So we see that same root word, kinesis. Kinesiology is the study of body movements. But here, cytokinesis means cell movement, and this is when the cytoplasm and the cell membrane of the parent cell split into two new daughter cells. So we call the original, the OG, the parent cell, and then we end up calling the new cells the daughter cells. And here's what it looks like. Slow motion. We'll see this process take place tomorrow, but you can see the very end of the cells splitting apart and starting to move away from one another. One more time. And there they go, moving away. Okay, so let's put it all together now that we know what happens in each phase. Again, we've got three phases, of the cell cycle: interphase, M phase, and cytokinesis. Interphase is by far the longest phase. It's got G1, S, synthesis, which is when the DNA is replicated, and then G2, which is another period of growth and preparation for cell division. Then there's mitosis, and then cytokinesis. And then quickly, some cells will re-enter this cell cycle and start going through it again. Now, another thing to point out here that we haven't yet talked about are these red bands. These red bands represent checkpoints. Keep that in mind. One important and unfortunate aspect of the cell cycle, one unfortunate outcome of the cell cycle is cancer. Cancer is defined as an unregulated growth of abnormal cells. Unregulated growth. So there's no control. You can't control how quickly or how randomly they are growing and dividing. And these are abnormal cells. So in this case, we can see what appears to be an endoscopic image of someone's probably large intestine. So this is probably colon cancer. It's a tumor that is growing up. This is a relatively small tumor that could probably be excised pretty easily through surgery, but this person is likely also going to have to go through chemotherapy in order to kill off the cancerous cells. That way they can't come back and start dividing again and potentially spread to other parts of the body. So there's another question at the bottom here. I want you all to tell me, what do you think causes this unregulated cell division to occur? And I'm going to go back really quickly just to point out these checkpoints that I mentioned. So what do you think causes this unregulated cell division to occur? Any thoughts? Okay. Well, no thoughts, then the answer is basically right here. We need those checkpoints in order to regulate the cell cycle and to prevent mistakes. These checkpoints allow the cell to stop growing and stop dividing if necessary. So if something goes wrong, we need these checkpoints in place to say, hey, you are not in a position to go on and divide and create more copies of yourself. You're messed up. Your DNA has not been replicated properly, or you've got too many mitochondria. Something is wrong with you. So we cannot allow you to divide. Your chromosomes are misaligned, your DNA has been damaged, something is not right. So we cannot allow you to continue to divide. At this point, our body has specific defenses that tell these messed up cells to die. We signal them, okay, you've been damaged in some way. It's time for you to die so you can be replaced by a healthy cell. Cancer occurs when these checkpoints are not working, when cells that have been damaged are allowed to continue to divide. And now they start dividing rapidly and beyond control. So we need good checkpoints in place to prevent cells that are already damaged from dividing. All right. Oops, this is not what I want. Well, yeah, this is basically what I want. So you do have an assignment called Cell Cycle Practice. It is on Canvas. We've got 15 minutes left in class. That's probably actually enough time to complete the assignment. Go ahead and knock it out so you don't have to worry about it anymore. I'll be here on mute if you have any questions. Good job today. I appreciate the contributions and participation. It's now 11:34. So, Sade, you asked me this a few times every week, but we in class at 11:35. So what I wanted to say something before you all go, which is that I sent a link into the chat, and that is for folks who have not yet completed their exam. You need to fill this link. You need to go to that link and schedule a time to do it. There were 8 of you who did not submit your exam. I am updating gradebooks this evening. So if you have not submitted the test by this evening, you will see a 0 in the gradebook, and it will hurt your grade pretty dramatically. So I need you guys to make sure that you're doing that, taking that seriously, so we can find a time to get that test taken. All right, it is now 11:35, so now you can go. Enjoy your day, and I'll talk to you all tomorrow. Did you have a question, Gloria?